everyone. My presentation is titled Combining RNA and DNA short sequence reads to assemble the mitochondrial genomes of flatworm parasite of fish. For this, I use Monogeneans. Monogeneans are parasitic flatworms economically important because they can cause disease in farm fish. Although Monogenea is a highly diverse group, approximately 3,000 species, mitochondrial genomes, mitogenomics, have been reported for approximately 37 species. Therefore, the goal of the presented study was to assemble and annotate the mitogenome of two monogenean species by using next-generation sequencing short reads obtained from low-input protocols of DNA and RNA. The DNA and RNA were extracted using the kit DNA RNA mini-prep kit. DNA and cDNA was submitted to enrichment using the Replicate single cell kit and Smart Sequencing kit. The sequencing was run in NextSeq 500 Illumina platform. Pre-assemblies were obtained from DNA sequenced with megahit and CAP3 assemblers, and from cDNA sequenced with Trinity, megahit, and CAP3. The pre-assembled were aligned with Mega to produce the final assembly. Gene prediction and annotation was obtained with RFFinder and Arwen. For the phylogenomic analysis, mitogenome from other monogenes were used, and the math PhyloSuite partition finders, RAG-ML, and Mr. Valles program were used. The complete mitogenome of Rhabdoxinocus viridis, which has 36 genes, was assembled. In the graph, it can be seen how the assembly using the cDNA helped to find regions without coverage from the sampling using DNA. In the same way, the mitogenome of Scutohis longicornis was assembled. The phylogenomic analysis allowed what positions the two species studied within Monogeneans, and specific gene arrangements were found for certain families of Monogeneans. The present study yields the complete mitogenomes for two species of Monogeneans, which was possible through the use of Illumina short-read sequencing, reamplification techniques, and integration of RNA sequence data. The greater coverage of certain regions in the mitochondrial genome from the cDNA reads, together with the use of megahit and Trinity for their assembly, allow us to obtain all the genes in the mitogenomes.